Hello everybody and welcome to this presentation. The research line showed here is application of NMR for food and beverage analysis, metabolites evolution and differentiation. In particular, we work in three areas. The first area corresponds to evolution and differentiation of wines. We have studied by NMR the evolution of different wine metabolisms different stages such as alcoholic fermentation, malolactic fermentation, and aging. The second area corresponds to evolution and differentiation of edible fat. Collaboration with the company Embutidos Luis Gil, the Spanish sausage production has been monitored by NMR. The third area corresponds to microbial susceptibility analysis. By NMR, we have carried out the calculate of this data. Specifically, in the field of enology, we will be assisted by the century-old research center, such as the Aro Enological Center. In the recent years, this center has incorporated a nuclear section. This methodology has the ability to discriminate, for example, grape varieties, countries, or regions of origin. Moreover, we have an intensive collaboration with Professor Søren Engelsen of the University of Copenhagen, an expert in NMR data processing. In summary, we offer the possibility of developing a multidisciplinary project in the field of NMR applied to food and beverage with a close relationship with the productive sector, such as wineries. Thank you for attention, and we contact me for anything.
Hello, my name is Ana González. I am the coordinator of the project's Plasma and Machine Learning Group at the University of La Rioja. Just let me introduce you my colleague Fernando Alba, who is going to talk about our cold plasma research line.
Cold plasma has a lot of application in agriculture, food industry, and cancer treatment. We use direct plasma to sanitize oak wine barrels. In this case, we obtained the complete removal of Brettanomyces from wood. As I told you, the energy of plasma could be transferred to water to create plasma-activated water. There is a lot of application of plasma-activated water in food industry and also a great number of methods to create plasma-activated water. Our research group have developed several ways to create plasma-activated water. The difference between these generation methods are related in which way we put in contact plasma with water. Cold plasma could be used to apply functional coatings over heat-sensitive surfaces. In this case, we have applied a two-layer coating over the stainless steel in order to reduce the biofilm produced by Listeria monocytogenes. Well, why is this research line important? Well, cold plasma is cheap, environmentally sustainable without using chemical and generating waste. It can be used with heat-sensitive surfaces like human skin, medical devices, food, and surfaces in contact with food. Which issues are we trying to improve? Well, we need to understand the decontamination mechanism, to transfer this technology to the industry, to promote regulation for safety use. For that, our research group participates in two cost actions of the European Union. Thank you very much.
I am Luis Moreno. I am the principal investigator of the GENUD research group, working mainly in pediatric nutrition, and the key topics of our research are nutrition, physical activity, obesity, mainly in children and adolescents, considering other aspects like growth, energy and food intake, genetics, and performing in vivo studies. The main topics of this research line are the risk factors of obesity development in children, the interaction between genetics and lifestyle factors, parenting practices related with diet and physical activity, new strategies for childhood obesity prevention, especially co-creation, and the impact of social vulnerability on obesity-related issues. And the relevance of this research line is justified by the elevated frequency of childhood obesity that is a hidden pandemic and is related with major non-communicable diseases like type 2 diabetes, hypertension, or cardiovascular disease. There is also a major economic cost of obesity care, the difficulty to treat children and adolescents with obesity, and this allow us to recommend prevention as the first option. We have developed a model of a study of obesity in children, considering different critical periods. The first one from conception until the end of the second year of life, the second around the development of the adiposity rebound, and the third one related with puberty and adolescence. And we have been working in an international network, especially in Europe, because we have participated in different projects, and the most recent ones are DigiCare4U and Grow.
The Plant Physiology and Agrobiology group of the Public University of Navarra is composed of around 20 members. Half of them are university professors, and the rest of students are other collaborators. We are focused on understanding how plants interact with the environment from a molecular physiology perspective. And our main research line is focused on nitrogen fixation in legumes, a research line which is led by myself and Estíbaliz Larrainzar, who is a Ramón y Cajal fellow. Our main interest for last years has been focused on the carbon-nitrogen interaction in legume nodules, and also how the plants interact with the environment. Now, our main interest is related to the role of ethylene in the regulation of the legume rhizobia symbiosis, a critical issue to understand how legume plants can better contribute to a more sustainable agriculture. Using tools of molecular biology, plant physiology, microscopy, etc. The project will be carried out at the Public University of Navarra with the collaboration of the company Blue Agro Bioscience and two universities in France and Austria. We can provide a friendly work environment, and if you have any questions, please do not hesitate to contact me for further information.
Hi, my name is Jordi Recasens. I am the coordinator of Weed Science and Plant Ecology Research Group from University of Lleida and Agrotechnion Center. We are working on sustainable agricultural practices for weed control, and our specific research line are biological processes that identify the harmfulness of weeds, establishment of integrated weed management programs, the characterization and control of herbicide-resistant biotypes. We study the ecological processes associated to invasive weeds, and also we establish weed management programs in vineyards by cover crop and mulches. The relevance of integrated weed management are according with different reasons. First, weeds cause overall more yield losses than other pests. By other hand, there is a great increase of herbicide-resistant weeds in different crops. There is also a continuous cases of weed invasions in agricultural systems, and apart from the effect of herbicides, tillage and mechanical weed control generate a higher carbon footprint. For these reasons, the specific scientific objectives of our research are the identification of new cases of herbicide-resistant weeds in maize and vineyards, with a specific emphasis in invasive species. Also, propose integrated weed management programs for these herbicide-resistant weeds, and establish in ecological vineyards alternative weed control methods to enroll tiller by cover crop and mulches. For these reasons, all people interested in our group will be welcome to work with us. Thank you very much.
Hello, I'm Roberto Soliva, professor at the Department of Food Technology, University of Lleida, member of Campus Iberus. This is to introduce you to one of our research lines in the sub-area of food science and technology, dealing with the development of food processing technologies, novel food processing technologies. Our group has a long track on the development of these technologies, with a focus on non-thermal technologies such as pulse electric fields and pulse light, among others. Our research works normally are normally focused on the interactions between food processing, composition, and structure and functionality in order to increase quality and the health-related effects of foods. Another research topic within our activity is the characterization and valorization of byproducts, especially those from the industrialization of fruit and vegetables that are rich in dietary fiber. Here we are working with extracts and the use of advanced technologies to obtain these extracts and apply them in the formulation of food products. These research topics are very timely nowadays, as we as consumers are demanding products that are not only safe but also tasty and appealing while being produced in a sustainable way, and at the same time allow us to follow a healthy diet. So our research generally involves the evaluation of these interactions between processing and food composition and structure that are relevant to quality and shelf life. Researchers who enroll our program are committed to evaluate the application of non-thermal technologies for the development of plant-based foods with this aim. So our focus is broad, very broad. We are addressing aspects that may be related either to safety risks, to quality properties and related compounds, or to food functionality, composition, and structure. So thank you for your attention. For more information, please contact us.
Hi, I am Gustavo Slaffer, the head of the Crop Physiology Lab. The lab is formed by crop physiology and crop improvement areas, focus on understanding crop yield and quality. The focus of the group is identifying major physiological attributes and genetic bases, determining yield in wheat or barley, studying plasticity of traits, and the work is translational in nature. In the past, breeding has improved yield through improving grain number and improved grain number through improving spike weight and anthesis through introducing semi-dwarfing genes producing plant height. In the future, we need to keep increasing spike weight and anthesis, but without further reducing plant height. And we can also try to improve fruiting efficiency, a trait related to floral development that we were working with in the last several years. The work done by the group has been not only prolific, but also receiving a high recognition from the international scientific community, with more than 1,000 references made to our papers per year in the last 3 years. The group is very much international. We have PhD students coming from many different countries in the world, and also our graduate students, when they finish the PhD, are working in many different countries, and we have research collaborators in projects or doing other kinds of collaborations in all continents. So thank you for watching this presentation, and we are looking forward to working with you. Thanks.
Hello. The group of environmental physical chemistry gather chemists, physicists, agricultural engineers, etc. And currently people come from Spain, Ecuador, Argentina, etc. We are interested in the distribution, transformations, and physiological effects of nutrients such as phosphorus and also toxicants. We want to understand the global cycling of these nutrients and toxicants in natural waters and soils. We use electroanalytical techniques such as Agnes. We also use the donor membrane technique. This is an example of one deployment. We also use the technique DCT, which now we want to apply intensively to soils in order to follow fertilization processes. Our work includes physicochemical modeling, numerical codes, field work, but mostly work in the lab. We have a large number of scientific collaboration all over the world. Thank you.
The group of the Agronomy and Environment in Mediterranean Agricultural System is composed by several staff located in the University of Lleida. The main objective is the development of appropriate agricultural management practices for improving crop productivity and enhance the adequate use of the natural resources as soil and water. The main topics of the research are the optimization of cropping technology and the analysis of the carbon and nitrogen dynamics and other crop nutrients. This research is important because it provides integrated knowledge for the agronomic, environment, and economical aspect of the cropping technology. We are trying to solve the aspect of the sustainability of the agricultural production systems for feed the population in the coming decades.
Hello, I would like to discuss a little bit our research group and the main lines of research that we are currently undertaking. So we are a group composed from people from the University of Zaragoza, the Hospital Servet and the ITA, and we focus on the development of new methodologies for trace elemental and isotopic analysis. We have many years of expertise with a number of atomic techniques based mostly on atomic absorption or ICP-MS. And we have a current focus on biomedical application and on analysis. I don't have the time to discuss everything that we do, so I will focus on one particular example. It's minimally invasive analysis using what is called as dry blood spots. This is related with blood sampling. We all know what venipuncture looks like and advantages and disadvantages. But alternative has been explored for many years, and perhaps the most successful one consists of depositing a few droplets of blood onto filter paper. This is very interesting, not only for children, but also because this material is quite stable. You don't need refrigerators, and also enables to develop home collection schemes. And this is something we're particularly interested. We believe that this scheme of preparing your own sample and sending to the lab could be appropriate in a variety of situations where elemental and isotopic analysis is key. We're talking about screening of toxic metals. We're talking about early detection of some diseases associated with the levels of metals, or we're talking about monitoring of chronic patients. And basically what we do is to test new specimens to produce the DBS with a known volume, easy to use for the patient, and then we develop the methodologies for the analysis. If you are interested, we have more information in this website, or you can contact me for further discussion.